All right, we can get started. So it's my pleasure today to introduce Professor Marinka Zitnik. So she is an assistant professor at DBMI here at Harvard Medical School. And she will be talking today along with her grad student, Michelle Lee, who is a PhD candidate in the Bioinformatics and Integrative Genomics program about functional AI for genomic medicine and therapeutic design. And so we are so excited to hear from you, Marinka. And without further ado, I'll turn it over to you.
Great. Thank you so much, Jesslyn, for this kind introduction, and to Sunil and Robert for giving us opportunity to speak today about the recent advances in AI for genomic medicine and therapeutic design. And throughout this talk that I will share with my wonderful stellar PhD student, Michelle, we will highlight several projects, those projects have associated data sets and databases that are all listed on our lab website, and you are encouraged to check them out. So without further ado, let's begin. Artificial intelligence is being increasingly integrated into scientific discovery in order to augment and accelerate medical research. And we have seen early successes of how it can help scientists generate hypotheses, design experiments, collect and interpret large quantities of data, as well as gain new insights that might not have been possible using traditional scientific methods alone. In areas of genomic medicine and drug design, there are several of the big advances that happened over the last few years that have created fundamentally new opportunities. And this includes the ability to predict three-dimensional protein structure from primary amino acid sequence, the ability to perform large computational screens of chemical libraries in order to identify what are drug-like compounds that have certain acceptable safety or efficacy profiles, as well as various other algorithms that can help us identify and nominate candidate therapeutic targets, model their binding to novel drugs, or even generate new designs such as small molecule compounds or protein pockets in order to identify and guide the process of drug design and development. So, and many of these research directions are really contingent or motivated by the fundamental observation that the space, in particular the chemical space of drug-like molecules, is incredibly large. It's so large that we are unlikely to be able to ever exhaustively enumerate and test it all. And so within this massive space of chemicals, the number of FDA-approved therapies and compounds is really small. We go from the range of 10 to the power of 60, which is an estimate for the number of drug-like compounds that potentially exist, to only around 10 to the power of 5 chemical compounds that have been approved as FDA therapies. Nevertheless, we have seen just incredible progress in medicine over the last century. So imagine what could be possible if we could put to use advanced computational methods and artificial intelligence methods in particular, in order to expand this tiny green point that I'm showing here on this slide, and identify what are other molecules that might be potential candidate therapeutics. So can AI really help us accelerate and augment the design of safe and effective therapies? And so I want to answer this question in this talk and by illustrating two key capabilities of a new generation of AI methods that we have seen being developed over the last few years, but mainly focusing on the last year. And so in the first part of the talk, I will speak a bit about AI foundation models, in particularly with the goal of designing general purpose models that can process a variety of prediction tasks in a unified formulation. And in the second part of the talk today, Michelle will present her recent research on contextual AI models that enable models to generate predictions across diverse biological contexts such as diseases and cells, in an effort to make predictions more actionable, meaning that they can more readily translate to downstream experimental investigation. So with that in mind, let's start with that first part on general purpose AI models, and in particular, what are the therapeutic tasks that these models can consider. And so the work that we are doing in my group within that area is generally released and made available to Therapeutic Data Commons, which is an initiative that sits in between biomedical scientific community and AI and machine learning community, and serves as a meeting point between biomedical scientists and AI scientists. And Therapeutic Data Commons really, at its core, provides reliable data sets, pre-trained AI models, and ability to generate scientific hypotheses with the goal of facilitating algorithmic innovation, as well as supporting advances in therapeutic science. And so generally how it works is that we start with some set or class or group of candidate therapeutics, and within that space, with that candidate therapeutics in mind, within Therapeutic Data Commons, we leverage a large number of AI models that are trained on publicly available data repositories. And those models are then applied to candidate therapeutics, and they optimize them for safety and efficacy endpoints in order to provide better scientific hypotheses that can then lead to laboratory and clinical evaluation data, and that data can be integrated into the models for continual improvement. Very broadly, this ecosystem has 4 key components. And so the 4 key components that this ecosystem has in order to evaluate AI models across therapeutic modalities and stages of discovery are the following. So at its core, we have curated benchmarks and benchmark data sets and mathematical definitions of AI tasks that are accessible to machine learning scientists, who can then incorporate those tasks and start addressing them by developing new algorithms. Those algorithms and their performance is reliably benchmarked and presented to the community through leaderboards that show best-in-class methods for individual therapeutic tasks. And beyond that, we argue that it's not sufficient anymore to simply be providing process data sets and standardized methods for model evaluation, but with the increase in availability and development of large AI models, it's important to start sharing also large models and their model weights, not only data sets used to train those models. And so the third big component in Therapeutic Data Commons are these pre-trained models that are designed for key therapeutic tasks that allow then users to interact with underlying therapeutic predictors in natural language, or even interact with them through interactive interfaces and designs that can facilitate collecting users' feedback. And so we have seen over the last 2 years many uses of the commons for a variety of tasks throughout therapeutic discovery. And those include tasks in early drug design, including molecular property predictions and ADMET optimization, modeling of geometric structure and binding of novel drugs to candidate therapeutic targets, as well as optimizing molecules in both local and global sense to change their design and change their structural properties that would maximize the profile of the molecule towards being most acceptable and desirable for a specific downstream endpoint. And of those many use cases, I will focus today on one, which is that of therapeutic use prediction. And I will walk you through a model that we have developed and how it integrates into TDC and what its key capabilities are. And so the motivation for that is really that we are seeing an increasing interest in developing large AI models for including for therapeutic science, where we have seen these large AI models being developed for areas outside of medical research and science most recently. But what the promise of these large models is that scaling models to very large data sets, we expect those models to have much better generalization capabilities than models that were trained in the past. So in biology and medicine, we see biomedical data collected on diverse entities, and they are recorded through diverse types of modalities. And so it's non-trivial and quite challenging of how to train large models on data at scale where the underlying data is so diverse and heterogeneous, and do that in such a way that when a model is trained, it can then solve new tasks. And we often want those models to solve tasks in a zero-shot manner, meaning that it can answer questions such as those here listed on the right part of the slide without having access to large or even any labeled data point for that tasks. And so the model that we have recently developed that has these kind of capabilities is called TXGNN. TXGNN stands for Therapeutics Graph Neural Networks. And it's a model that is trained on a comprehensive knowledge graph that covers 70,000 clinically recognized diseases. And that knowledge graph represents a consistent, unified data set that brings together these diverse biomedical data modalities. And once trained, TXGNN can process various therapeutic tasks and predict and candidate indications and contraindications across this entire range of diseases, and it can do so in a unified formulation. So let me explain what TXGNN and how it operates and what are key components of this model. So fundamentally, the way TXGNN makes predictions around individual disease and a therapeutic candidate is that it models the effect of the therapeutic effect by directly encoding information on the primary targets of a therapeutic candidate, as well as how those targets interact with each other through the underlying biological network, and where in that network are those genes that are associated with the disease that the therapeutic is considered for. And so to model this view, we ground the TXGNN model in known disease mechanisms and known treatment effects across the entire range of FDA-approved therapies and a range of diseases with some available treatments. And so this graph machine learning model, TXGNN, is then trained on this large knowledge graph. And once trained, it can answer queries around new drug-disease relationships. So what is this knowledge graph that the model is trained on? This knowledge graph is prime KG knowledge graph that we have built over the period of 2 years, and it's one of the largest knowledge graphs in terms of having a really broad coverage of human diseases. That was our key motivation and focus in developing this knowledge graph. It's broadly collated and constructed based on 17 biorepositories, and it contains diverse kinds of biomedically meaningful types of relations and additional information and attributes of those biomedical relations. This knowledge graph is publicly available. We host it on Harvard Dataverse, as well as we provide Python scripts that allow anyone to update the knowledge graph as new data become available. So it can be continually updated and so that it remains up to date for future analysis. So given this knowledge graph, we want to then design a model that can perform well across a broad range of diseases, right? That was our motivation, and that's what's required if you want to have a broadly useful model. And so in existing research, there are several strategies that are established and commonly used to evaluate models in this area. And so most models are of the kind that they start with the ground truth data set of drug disease pairs or drug disease indications and contraindications, and then split them in the random manner. And part of those drug disease indications are in the training set and the rest of them are in the test set. And then we want the model to perform well on those unseen indications that are in the test set that the model does not encounter during training time. And we will refer that to scenario A. And the critical characteristics of this scenario and this strategy is that by splitting this data, drug and diseases in a purely randomized manner, it quite often happens that these diseases that the model encounters at test time are similar to diseases that the model has encountered in training time. And very often diseases at test time might have some known treatments and potentially rich molecular underpinnings. And so what we are seeing that under this setting, many existing methods already perform quite well. And so what I'm showing here on the right part of the slide is performance of a standard graph neural network measured as area under precision recall curve. So higher value indicates better performance. And we can see that under this regime, where this kind of diseases that the model is asked to reason about at test time are diseases the model has already seen during training time. In this setting, the model, many models and many established methods already perform really well. So we're not so interested in those scenarios, in scenario A. We are more interested in scenario B. And so that's a scenario where we encourage the model to perform well on diseases that have no or very few existing treatments, that have very poorly characterized mechanisms. And we want the model to identify candidate therapeutics for those diseases and for those disease areas. And kind of technically, the way this is seen and manifest in the knowledge graph is that for those challenging to predict diseases, they tend to have really sparse neighborhoods in the knowledge graph, which is shown here in the scenario B part of the slide, where this central blue triangle represents a disease and disease node. And we can see that the surrounding local neighborhood around this blue triangle is really, really sparse, meaning that there are no links between this disease and any other drug node, that potentially this disease has poorly understood molecular mechanism. And so it becomes really challenging for existing methods to perform well in those settings. And this is not just speculation. We can really evaluate a state-of-the-art models under the scenario B, where the model is tasked at test time to predict and reason about the diseases with no treatments that could not be easily modeled. And we can see that performance really drops drastically. So the natural question that emerges is, can we train AI models on data at scale, in particularly leveraging advances in AI that include self-supervision and transfer learning, and make those AI models work well on diseases with no existing treatments, which effectively means that we want models to perform well on zero-shot tasks, where those tasks are diseases with zero and known treatments. And so that's exactly what TXGNN has been designed for. So it's a graph neural network, which is a type of model that falls under the broad scope of geometric deep learning models, that starts in this left bottom part of the slide, which is with a neural message passing strategy trained on the knowledge graph. And the purpose of that neural message passing strategy is for the neural network to learn what is effective way to propagate latent representations or embeddings along edges of those graphs. And those are called neural messages in default, in such a way that this process will result in really powerful, meaningful representations of diseases and drugs that one can then leverage in the latent space to answer queries around diseases and drugs. And the way TXGNN can achieve that is by using neural message passing on the knowledge graph, as well as relying on a novel transfer learning mechanism that allowed the model to transfer information from diseases that are better characterized to diseases that are poorly characterized and have no known treatments. And this is done through the process of gating, that is a mechanism that can combine two sources of information, which one is information that is native to every disease, and the other source of information is information transferred from other diseases. Once this latent representation space of embeddings is learned, then in the fine-tuning stage, the model can be directly used for prediction around novel diseases that it has never encountered during training time, and it does not require any labeled drug or any known treatment for those diseases in order to make prediction. So let's see how TXGNN performs. So once we train TXGNN model, the key capability that we want this model to have is to be able to perform zero-shot inference on a broad range of diseases. And that's our primary mode of evaluation. So the way to read the right plot on this slide is the following. On the y-axis, we show area under precision recall curve, which is performance metric, and higher indicates better performance. And then we have results shown under scenario A and scenario B. So scenario A, just as a recall, you know, this is the scenario where the model only is asked to reason about diseases which already have some known treatments, known molecular understanding, and those are diseases that are easy to predict. And we see some lift in performance in TXGNN model over GNN model, other GNN models, and we are quite happy with that. The part that really is excited is that in scenario B, where the model is asked to predict, make predictions around novel diseases in a zero-shot manner, we see this lift in performance that is really substantial over prior work. And in particular, TXGNN improves over existing methods by up to 49% in accuracy when predicting indications, so positive use of therapies, and over 35% and achieves over 35% higher accuracy when it reasons about contraindications. And what these plots here on this slide show is when the performance of the model when we break it down by different disease areas, meaning that when we put all diseases within a specific disease area, such as adrenal gland or cardiovascular diseases or cancer and cell proliferation diseases in the test set, none of which the model has ever seen, and then we ask the model to nominate potential indications and potential contraindications for those diseases. And here performance is measured as a recall at 100, where we are calculating out of top 100 predictions that the model made, how many of those top 100 predictions are actually correct. And we're seeing substantial lift in performance across the disease areas relative to prior state-of-the-art methods and non-guided strategies. Of course, the next natural question to ask is, what about novel predictions that the model would make? So here we ask the model to make novel predictions, and so those are predictions around candidate indications and contraindications that are not yet approved by FDA. And we took those novel predictions and evaluated them against off-label prescription decisions made by clinicians in a large healthcare system. So here we relied on our collaboration with teams from Mount Sinai Hospital in New York City. They have spent many years of research working with their EHR system and phenotyping the data from their EHR database to derive then the natural experiments where patients were put on a certain drug for having a disease and then put on a secondary drug in an off-label manner. And then we asked whether there are novel predictions made by the model that the model is very confident about. So top 1 or top 5 most highly ranked predictions made by the model for every disease. Do we see those predictions enriched in off-label prescriptions? And the answer to that is yes, we see those novel predictions being consistent with off-label prescription decisions made by clinicians. Another point of analysis that we can do is, because we have the ability to generate the knowledge graph at different points in time, then we can essentially generate the knowledge graph, freeze it in time, and then ask the model to make predictions that will later evaluate against more recent FDA approvals. And we have done that for numerous time points to indicate and simulate what kind of predictions the model would provide if it would be used in a prospective manner. one important component in developing these broadly generalizable models is to think about how to make models accessible to end users. So with that in mind, we have designed a clinician-centered AI design, which is an interface that can support scientists in interacting with AI predictions and also interpreting conclusions of AI analysis provided by TXGNN model. So this is available online. You can go and check it out at txgnn.org. And the way it works is that the users can enter their disease of interest or the drug of interest, and they get predictions and ranked lists of predicted drugs for that disease produced by TXGNN. And they can explore the latent embedding space of drugs and diseases learned by the model. And they can also visualize reasoning paths or explanations that are paths in the knowledge graph, starting at drug, ending at the disease, that traverse different types of biomedical entities that the model has relied on when making a prediction. And so the reason why we call this clinician-centered design is because not only we design this user interface, but we wanted to understand what is the usability of this user interface and what's utility of these explanations for scientists making decisions and reasoning about hypotheses provided by AI models. So we have done a usability study of TXGNN with a panel of clinicians at Mount Sinai, and we compared TXGNN model to a baseline that had no explanations in terms of human user accuracy, exploration time, which would be measured as the amount of time that an end user had to invest into understanding or deciding whether this prediction made by the model is worthwhile exploring further or not, the confidence that they have in model predictions, and the agreement with model predictions. And we have done that across a spectrum of usability questions. So what that analysis showed is that not only it's important to include different ways to provide insight into model predictions, such as visualizing latent space and providing explanations, but in fact, the way those explanations are presented to the user, such as in the form of paths or in the form of subgraphs or nodes, also plays a vital role in deciding whether a particular tool will be useful to users. So to summarize this model, that is trained on the entire data set across 17,000 diseases, is now publicly available at txgnn.org. You're more than invited to try it out. This would not also be possible without a really fruitful and exciting collaboration we have had with Niels Gellenberg's lab from the DBMI department, that brought where he brought the expertise in the design of these models, as well as some of the usability studies. We have several follow-up studies into specific disease areas to evaluate these models. Another point that I will make, in particularly of what is next beyond developing these kind of models that can provide predictions across a large number of disease phenotypes. Well, I think that the next frontier is to be able to provide universal predictions across disease states and phenotypes that would not be limited to a priori defined medical vocabularies, where we generally limit model predictions to diseases and drugs that are defined and exist into vocabularies such as ICD codes or human phenotype ontology or gene ontology or drug bank and others. And so the way this is possible nowadays is to leverage large language models to essentially design a version of these interfaces that can enable direct communication between human experts and AI models. And that is important for two key reasons. First, it can allow us to support any disease-relevant phenotype without limiting ourselves to a priori defined phenotypes from a medical ontology or vocabulary. And second, if this communication is direct, then it's possible to continually refine AI-generated hypotheses in similar ways as I'm sure you tried out interacting with ChatGPT and similar tools, and then continually refining, in many cases, responses and suggestions that those tools provide. So a version of the model that we are developing now, it's called TXPLM, which is a therapeutics-oriented protein language model. It's an architecture that has two key components. So it has the backbone language model that is trained on protein sequences that captures biological knowledge, and a front-end language model trained on natural language that allows end users to come and describe their pathway and disease of interest by just providing a paragraph in English language. And that paragraph is then mapped to the latent space together with the embeddings produced by the protein language model to prioritize proteins and domains. And we're working with several groups at MIT and CZI on validating prioritized proteins and protein domains for as candidate therapeutics and validating them through flow cytometry experiments. So this concludes the first part of the talk, which was focused around the opportunities created by new generation of models in therapeutic science, with a particular focus on models that can handle a large number of diverse tasks simultaneously. And I described TXGNN as an illustration of the model that goes in this direction, and serves as a model that can handle a variety of indication and contraindication tasks in a unified formulation. And now in the second part of the talk, Michelle will kind of switch the topic a bit, but only a bit, and she will talk about molecular cell atlases and ways those can be leveraged to contextualize some of these models. Okay, let me stop sharing the screen, and Michelle, take it away.
All right, thank you so much. Thank you, Marinka. Thank you, everyone. I'm Michelle. I'm a PhD candidate in the BIG program. And as Marinka mentioned, in the second half of the seminar, I'll talk about contextual AI models and their ability and potential to enable prediction across biological contexts, such as diseases and cells. So what do I mean by context, and why do we need it to understand genes or proteins or diseases or drugs? So let me answer you with an analogy. What do words have in common with proteins? Their meanings arise from their context. For example, if you take the word apple and Google it using two different sentences, do you think that Google will spit out the exact same suggestions? I would suggest no, because even though they are the same word, they have very different meanings depending on how they are used. On the left, apple is referring to the fruit, and on the right, apple is referring to the company. And the same is true for proteins. For example, LEF1's role varies depending on where it is in the human body. In the hypothalamus, LEF1 contributes to hypothalamus development. In the blood vessel, LEF1 is contributing to angiogenesis. And so context is incredibly important for characterizing proteins. And there's, of course, lots of research done on protein representation learning. Two types that I'll just mention here are structure-based and sequence-based. So structure-based methods take as input the 3D structure of a protein and output a low-dimensional vector representation or an embedding of the protein. Sequence-based methods take as input a sequence of amino acids and an output an embedding of the protein. And these embeddings can then be used for any downstream task. And these methods have led to incredible advances in drug development, precision medicine, and lots more, but their protein representations are context independent, as in there's only one embedding per protein. So now you may be wondering, well, what's the benefit of context-dependent protein representations? If we had context-dependent protein representations, we could improve the accuracy of predicting drug specificity, safety, and toxicity. For example, design drugs such that we can minimize off-target effects in different tissues or cell types. and enhance predictions of patient response to treatments with cell type dependencies. For example, modeling patient phenotypes alongside the affected cell types to better predict response to that particular drug. So our goal here is to develop a self-supervised geometric deep learning model that can generate protein representations in different cell type contexts. And unlike existing methods, which only generate one embedding per protein, totaling about less than 22,000 protein embeddings, our approach generates many embeddings per protein, totaling over 300,000 protein representations. And here we are assuming that one gene encodes one protein. So we're not considering any structural differences of protein isoforms or conformation differences. So the context that we care about here is the cell type context. And so here is AWARE, our self-supervised geometric deep learning model that can generate protein representations in different cell type contexts. So let me begin by explaining the illustration on the left. So to be able to model proteins across cell type contexts, of course, we need cell type specific protein interaction networks. To construct these cell type specific protein interaction networks, we start from a multi-organ single cell transcriptomic atlas of humans, which consists of samples from 15 donors, resulting in 483,000 cells after quality control processing. For each of these cell types that are annotated by the consortium, we get a list of genes that are activated in the cell type. In short, activated means that these genes have a higher average expression in the cells of interest compared to the rest of the cells in a data set. And from these lists of genes, I take the global reference interaction network, extract the proteins that correspond to the activated genes, and take the largest connected component. And as a result, each cell type has around 2,500 proteins on average. And so we have 156 cell type interaction networks that span 62 tissues of varying hierarchical scales. Now, to model the higher order organization of cell types and tissues, we need a network that's composed of tissues and cell types and their relationships with each other. So we have cell type nodes in green and tissue nodes in blue. These cell type nodes are given by tabula sapiens, and each cell type we know comes from a specific tissue according to the tabula sapiens metadata. So those tissue nodes are connected to the 156 cell type nodes. Cell type nodes and cell type edges mean that the cell types interact, and this is based on ligand receptor expression. Our tissue-to-tissue relationships are parent-child relationships. So we take the nodes that are corresponding to the tissues that samples were taken from and take their parents up until the root node. And altogether, we have a cell type and tissue network, which we'll call the metagraph. And with all of the networks together, we have 156 cell type specific protein interaction networks and one metagraph of cell types and tissues. And our algorithm, AWARE, is able to learn representations for every single node in these networks. Concretely, AWARE allows us to integrate multiple data modalities using deep learning, integrating single cell transcriptomics data with interactome data, as well as tissue hierarchy, and perform transfer learning across the different contexts from proteins to cell types to tissues, and as a result, produce multi-scale embedding space that contains nearly 400,000 protein embeddings, 156 cell type embeddings, and 62 tissue embeddings, which in comparison to prior methods, those only one condition of the protein is represented. And with these contextualized embeddings, we can then perform contextualized predictions. For example, given the cell type context and a candidate protein, can we predict whether or not it's a safe and effective target for a drug? So let me give you an intuition of the model. So given a network like the one at the top, AWARE embeds the protein, cell type, and tissue such that the protein embeddings from the same cell type are attracted to each other if those proteins share an edge. If they don't share an edge, their embeddings are repelled from each other. Protein embeddings from the same cell type are attracted to the corresponding cell type embedding and repelled from other cell type embeddings. Cell type embeddings of cell type nodes that share an edge are attracted to each other, otherwise they're repelled. And the same similarly is true for cell type and tissue nodes that share an edge. They are encouraged to be close by. Similarly, tissue nodes that share an edge are embedded nearby or far apart otherwise. So going a little bit more in depth, how do we learn embeddings for each node? So there are different levels of learning. We have learning at the protein level, at the cell type level, and at the tissue level. And we use something called an attention mechanism to do this. For those of you who may need a refresher, think of an attention mechanism as what would you pay attention to the most to achieve your goal. So if your eye is your model, you may be interested in reading a story. So your options are a newspaper, a blank stack of papers, coffee, notepad, or a book. So if you're interested in reading a story, perhaps you'll pay more attention to the book, and then next maybe a newspaper. And the least will probably be to the cup of coffee, unless you, of course, need the attention to read your book. So the same is true for our case. So we have protein level attention. So for each protein node, which of its protein neighbors are important for that protein. Similarly, we have metagraph level attention for each cell type node, which tissue and cell type neighbors are important. And we have an attention bridge where we propagate information between cell types and proteins representations. And so altogether, we have multiple levels of learning embeddings in order to capture the multiple scales of data that we have in a unified embedding space. And to go through the objective functions a little bit, we have three components. We have a protein level, cell type level, and tissue level component of our objective function. At the protein level, we perform link prediction on the protein-protein interactions. We also predict which cell type the protein is activated in. At the cell type level, again, we perform link prediction only considering the cell type edges and the cell type to tissue edges. Similar is true for the tissue level loss, where we consider the tissue-tissue relationships and the tissue-cell type relationships. For link prediction, we use as standard binary cross entropy. And for this one cell type classification task, we do central loss. Okay, that was a whirlwind of an explanation, but to recap, AWARE is a self-supervised geometric deep learning that can generate many representations for each protein based on their different cell type contexts. And we have nearly 400,000 protein embeddings for a total of 156 cell type contexts. So let's take a look at how good these representations are. So here's a visualization of the protein representation space. Again, there are 156 cell types in the metagraph, and on average, 2,500 proteins per cell type. So we're visualizing 400,000 points here. Each dot is a protein representation, which makes it a little bit difficult to see clearly. So let's just select a few cell types to look at. So here I'm showing the fibroblasts of the breast. On the top, on the middle, the B cells, and the bottom CD4 plus helper T cells. Note that each dot here is a protein representation. And of course, we can't visually say that it looks like the regions are meaningful. Let's quantify this. So we use a spatial enrichment scoring algorithm called SAFE. And we show here the distribution of the neighborhood enrichment scores that are produced by this SAFE algorithm for each cell type context. And here, all of the 156 cell type contexts are statistically significant, as in we show that the spatial regions in AWARE's embedding space are significantly enriched for protein representations or originating from the same cell type context. And this suggests that AWARE's contextualized protein representations self-organize meaningfully in the embedding space. And we also calculate the similarity of cell type specific protein representations from the same cell type versus from different cell types. So as you can see, cell type specific protein embeddings are more similar to each other if they are from the same cell type in dark purple compared to representations of proteins in other cell types in light purple. And this is what we would expect that the dark purple will be further to the right than the light purple, and the light purple should be further to the left. And this effect is no longer observed when we remove any notion of cell type context in a model, as in we remove this central loss and the metagraph that is encouraging the separation according to cell type and tissue organization. And only remaining is the cell type specific protein interaction networks and the link prediction component for protein interactions. And furthermore, we quantify how well can our embeddings reflect the tissue hierarchy. So here we're looking at the embedding distance of AWARE's 62 tissue embeddings as a function of tissue ontology distance. A well-performing model would give us a positive correlation, meaning that the farther away the nodes are according to the tissue hierarchy, the more dissimilar the tissue embeddings are. And this is exactly what we see here. And compared to a null distribution, we in fact see a positive correlation between the tissue ontology distance and the tissue embedding distance. So I hope I've convinced you that AWARE's representations seem to be context aware and reflect cellular and tissue organization. So one possible downstream task that we can adapt AWARE for is predicting the therapeutic potential of proteins at the cell type level. So given a set of cell type specific PPI networks, AWARE generates cell type specific protein embeddings, which we can then fine tune in a downstream model for our desired task. And the question is, given the protein of interest, is it a strong candidate for the therapeutic area that we care about? And so we will get a score for each protein in each cell type. And this actually allows us to further generate hypotheses regarding which cell type might be highly predictive for examining the therapeutic target. So first, let's very briefly discuss the data. So we're taking data from open targets, which contains information such as clinical trials, drug indications, drug targets, etc. So to define a therapeutic area, we start from a disease of interest and identify its descendants in disease ontology. And here we're interested in rheumatoid arthritis-related conditions and inflammatory bowel disease-related conditions. And we create a list of clinical trials for these diseases, specifically those with at least one completed clinical phase 2 or higher. And from this list, we collect a list of drugs and target proteins for these therapeutic areas. And in total, the list of candidate targets for our therapeutic area of interest are as follows. We construct positive examples by selecting proteins that are targeted by compounds that have at least completed one clinical trial of phase 2 or higher, treating the therapeutic area of interest. And what this means is that these proteins are deemed safe and somewhat efficacious for humans with the disease. And of course, we need negative examples. And we do that we construct those examples by selecting proteins that have not been studied for the therapeutic area, but yet have been targeted by at least one existing drug or compound. And so here's our model performance for rheumatoid arthritis-related conditions using our metric average precision recall at 5. And so each dot here is representing a single cell type context. And the gray dotted line is training a standard graph attention neural network on the global reference protein interaction network. And the brown dotted line is training a state-of-the-art convolutional neural network for integrating data modalities called Bionic. These two approaches in the dashed line are context agnostic, so they do not consider any sort of context in their models. So as you can see, many cell types outperform the global reference network as well as Bionic. And this suggests that studying the therapeutic potential of proteins may be more accurate when leveraging the cell type context of these proteins. And these rankings have been validated by an RA clinician as being appropriate and very exciting hypotheses that are produced by AWARE. We can also look into individual proteins that are already targeted by drugs. And here we examine the most and least predictive cell types to better understand the mechanism of action. I'm showing here IL-6R, which is a very commonly targeted protein for drugs indicated for RA. And we validated these top most predictive cell types, and I identified one particularly exciting cell type to be myeloid dendritic cells. We've independently also trained a model for IBD-related conditions, and the same metric is being used, as well as the same baseline approaches, benchmark approaches. And again, we see that the many cell types outperform the 2 cell type agnostic models. Again, the rankings have been thoroughly validated, and we can also take a look at the protein level predictions and identify the top 5 and bottom 5 most predictive cell types. Again, these have been validated, and we're very excited about the hypotheses produced by AWARE. So to recap, AWARE trains cell type specific protein representations, and we can fine tune it for a wide range of tasks. I just showed you an example regarding therapeutic potential of proteins, but they can certainly be used for other applications related to preventative medicine, disease diagnosis, therapeutic discovery, you name it. So here are the key takeaways of AWARE. AWARE is a flexible approach that can be used to generate context-aware representations on any sort of contextualized networks. Here we use contextualized protein interaction networks that are created from single-cell gene expression data to help resolve protein interactions using single-cell resolution. And AWARE is able to generate representations that can then be fine-tuned for downstream tasks that have cell type dependencies or action mechanisms. And I want to take a moment to show you a demo of AWARE. So if I were to switch screens, you should be able to see my browser. So we have hosted our method AWARE, the complete model for AWARE on Hugging Face, which you can already access. And you can take a look at our embeddings at 2 different levels, at the protein level and at the cell type level. So suppose you have your favorite protein. I've entered TNF just because it's great. And you can press submit. You'll wait for it to load. I've already preloaded for the sake of time. And you can see which cell types this protein is activated in, and the coordinates in the space so that you can look for it easily. You can also look at the cell type level predictions, where you can select cell types that you're interested in looking at, press submit, and now you can examine the embedding space of AWARE. You can see how many activated proteins there are, the safe scores from the safe algorithm that I mentioned, and more closely look at the proteins that are in each of these networks. All right. So, all right. And one final comment I will make is, looking ahead, I think it's important, as we invest so many resources into creating large-scale pre-trained models, that we share them publicly to encourage opportunities for continual improvement and the reuse of models. And we hope that we have set an example by releasing our pre-trained AWARE models on Hugging Face and facilitating the Therapeutic Commons, for example. And we're particularly excited as well about a future in which we can create interactive interfaces that enable human-in-the-loop for improving efficiency and accuracy of our models. And with that, I'll pass the baton back to Marinka.
Great, thank you, Michelle. And so let me wrap up this presentation. So today we were excited to share with you some of the new capabilities of models that have been developed very recently. And we highlighted 2 important directions. The first one is that on developing models that can generally address a larger number of diverse tasks and do that in a unified formulation without requiring labeled data for each and every of those tasks. And second is the notion of contextualizing models and model predictions in order to enable more accurate prediction across diverse biological contexts, which was illustrated in this seminar through this context defined by cellular types. And so with that, we'll wrap up the seminar for today. This has really been a fantastic and great work with Michelle and other students in my group. And many data sets and these code bases are all available from our lab website. And we're also very happy to take questions in the remaining time. Thank you.
Thank you so much, Marinka and Michelle. This is great work. So we'll open it to questions. There's one in the chat from Ludwig, but it looks like I was going to give Michelle an opportunity to expand on it if you would like to. I know I see you've answered it.
Yeah, thank you for your question. Yes, so the global PPI network is a union of high quality protein interaction networks such as BioGrid, the Human Reference Interactome, and String. Is there any, do you have a follow-up question in addition to that?
No, I think it's a great approach, and I really like the presentation. Of course, I was just wondering. I mean, constructing a global reference network is always difficult, right? No matter which species you are working at, and no matter you want to construct a gene regulatory network or PPI network, they are themselves context dependent, right? So they are constructed with certain experimental methods for certain cell lines. So a global reference network, unfortunately, remains an illusion, but I think it's a sensible approach, and it was very interesting. Thank you.
Yeah, absolutely. Thank you for your comment.
Hi, Michelle and Marika. Thank you for this presentation. Very nice. I have a question about your work on cell type specific embeddings, Michelle. So at some point you showed us that I think within the context of rheumatoid arthritis, you found this one type of immune cell that was specifically interesting to look at for future drug targets. And I was wondering there, maybe it's a bit more of a chemistry or a pharmaceutical question, but is it already possible to specifically target a single type of immune cells?
That's a great question. So we were cross-validating our predictions produced by AWARE according to single cell atlases created from patients with rheumatoid arthritis. So you're right that we were not validating it based on a drug that was specifically able to target a cell type of interest. But there were results from that analysis on the single cell RA specific atlas that, for example, this myeloid dendritic cell IL-6R specifically in myeloid dendritic cells were a promising candidate or a biomarker for patients with RA.
Okay, thank you.
Mm-hmm. All right, we have another question in the chat from Tyrone. Would it be correct to think of the nodes in the primer and the prime KG as parameters for the purposes of training? And how are you handling isoforms and where's PPI?
Yeah, so both are great questions. So yes, you can think of nodes in the prime KG knowledge graph as parameters or kind of initial data points that specify actually the neural architecture, because the neural architecture is such that parameters of a neural network are propagated or exchanged between nodes that are connected by edges indicating existing biomedical knowledge. And the second question is, how did you handle isoforms in AWARE's PPI? Great question. So at this time, the global reference interactome really comes from HURI, which is the Human Reference Interactome Network published by Luck et al. In Nature 2022. And for that network, we do not handle isoforms at all. We currently do not have and that goes beyond the scope of our ability to access the data, isoform level resolved protein-protein interactions at the systematic level that would allow us to train the models on beyond doing case studies, which is what we have been looking at. But we're hoping that more of that isoform level data becomes available in the future.
Would you then be able to find so since you're able to isolate like a neighborhood, or at least I assume that's what you're doing as part of the attention training right there, could
Yes.
then fine-tune the attention training just to select particular isoforms then in that case? Because it doesn't seem like you need to keep it for the kind of accuracies that you're getting. You don't really need every single isoform. You just need to fine-tune it for whatever.
Yes, we hope that in that regard, the attention mechanism would really be helpful. And right now, it's been incredibly useful to essentially upweight and downweight certain interactions across cell types, which can help us mitigate some of the issues with respect to the incompleteness of the interactome network, or this underlying context specificity, or the fact that these interactions were measured with very different experimental platforms, as I think was mentioned in the beginning of the Q&A.
So the attention mechanism is just looking at the neighborhood right there. It's not necessarily looking at the, or is that what it's doing? I'm not quite sure on the specifics on that.
So at each layer, attention mechanism is looking at the direct neighbors of every node in the graph.
Okay.
But the way when you have a multiple layers and to construct a deep network, that effectively means that if you have a neural net with k layers, there you are effectively convolving conformation over k hop neighborhoods. But in each layer, every node only convolving or upweighting and downweighting information over its direct neighborhoods.
Okay, that's good enough for now. Thank you.
Thank you.
Well, thank you again, Marinka and Michelle. And this is really great work. Yes, thank you, Radam. We will see you all next week.